DNA, or deoxyribonucleic acid, is a molecule which encodes the genetic information used in cells. It is a double-stranded molecule that stores genetic information, and it is coiled in a double helix structure, similar to that of a spiral staircase. SSBPs are single-stranded binding proteins, which keep DNA from forming a double helix structure again. It makes it easier for helicase to separate the double helix. Next up is DNA polymerase I. It proofreads the new DNA strand and replaces the RNA primer segment with DNA because it must be removed to form a complete DNA molecule.
DNA polymerase III adds the actual new DNA nucleotides with the help of the sliding DNA clamp, which allows DNA polymerase III to stay attached to the strand itself.
Topoisomerase helps throughout the whole process. It allows the DNA to stay loose when being separated and avoid being twisted too tight and supercoiling. DNTPs are deoxynucleoside triphosphates, also known as free nucleotides, added on in between to complete the strand. DNA is a polymer composed of two long chains of nucleotides, a monomer.
DNA contains a phosphate sugar backbone which acts like a railing to the spiral staircase, wherein the nitrogenous bases connect the two strands together, acting as the steps to the spiral staircase.
Next, we will walk you through how DNA is replicated. The first step is that the DNA double helix gets separated into two strands. During this process, the H+ bonds between the nitrogenous bases are broken and separated by the helicase enzyme, effectively splitting the double helix into two strands.
Each strand of the split double helix becomes a parent strand for new DNA. For this reason, DNA replication is known as being semi-conservative, since the two copies of DNA are produced and each copy contains one of the original strands and one new strand.
The separation of the double helix creates a structure called the replication fork. The replication fork is a structure that forms during the DNA replication, which is created by helicase. The leading strand starts with the 5' end at the center of the replication fork, extending to the 3' end away from the replication fork, whereas the lagging strand starts from the 3' end at the center of the replication fork, extending to the 5' end away from the replication fork.
In general, DNA has two complementary strands, meaning one strand runs in the opposite direction to the other. In other words, one strand will go from 5' to 3', while the other will go from 3' to 5'. Because the strands run parallel to each other but with opposite alignments, DNA is called anti-parallel.
Because DNA is so long and compressed, in order to replicate it in a timely fashion, it must be replicated at various spots in the strand. These spots are called origins of replication, areas beginning in the middle of the strand and replicating in both outward directions.
Along with helicase, single-stranded binding proteins work to keep the DNA helix from snapping back together by coating the unwound strands with subunits of single-stranded binding proteins. This lets the DNA helix remain loose during replication.
Now let's talk about how DNA replicates on the leading strand. Replication is much easier on the strand and more continuous because polymerase I and polymerase III only work on the 5' to 3' direction. The complementary leading strand moves in the 5' to 3' direction as well, which makes it much easier for this process to be executed.
Because of this, RNA primase is easily able to apply RNA primer to the new strand, and polymerase III can simply add new nucleotides as the strand comes along. Polymerase I replaces the RNA primer with DNA to form a complete DNA molecule.
However, the lagging strand, which moves in the 3' to 5' direction, cannot replicate the same way the leading strand can. In this type of replication, the enzyme moves away from the fork, and the fork is uncovering new DNA that needs to be replicated.
Because of this, the lagging strand replicates in small segments called Okazaki fragments, synthesized in the 5' to 3' direction away from the replication fork.
First, RNA primer is applied through RNA primase. Then, DNA polymerase III elongates the sequence. After that, DNA polymerase I proofreads the sequence and then removes the RNA primer. Lastly, these fragments are then stitched together by DNA ligase, creating a continuous strand.